is that the genome is what we call unstable. The genome breaks and rearranges. It does enormous, thousands and thousands of things are happening. Okay, and so all of a sudden you have an enormous wealth of new information present in the cell, and you can't tell from among those thousands of things which are the ones responsible for the cancer and which are simply fortuitous because the whole genome is being jumbled. Okay. In sarcomas, that doesn't happen. So for us, for sarcomas, if we want to identify, say, 5 events that have occurred, it's much easier because you don't have a background of a whole myriad of other things happening that would confuse the matter as you do in most carcinomas. And for that reason, by looking at sarcomas and looking at several, we hope to start to develop essentially themes which then may be useful actually for carcinomas because it will be simpler to filter it all out. So I've already mentioned chromosomal translocation, breaking through chromosome, rejoining them. So the one thing you can say, well, why don't you model it by generating that chromosomal translocation? That would be the ideal thing. And the reason for that is, remember, you're taking 2 chromosomes, breaking each of them, and then switching partners. So in actuality, you don't have one thing happening, you have 4 things happening. You have 2 new breaks that are joining together, and you're losing the things that are participating, 2 things that are participating in making those breaks. Okay, so that's actually 4 things. And in the translocation, you generate all 4 things at once. Okay, when we're modeling in the absence of translocation, what we do is normally do one thing and then ask what happens with that and then do the other. But sometimes, and in many cancers, it will probably be important that all 4 things are present at once. Okay, but we can't quite model it that way. We've just recently developed, since I've made these slides, developed a technology to do translocations and to make any translocation we want and to do it at high frequency. But that's going to be the next generation of models. So instead, we're going to model it slightly differently, and I'll explain how that is. Okay, the cancer that we're going to talk about is called synovial sarcoma. And what was known about synovial sarcoma is shown in this slide, and that is that they occur near joints. Okay, and so, for example, at the knee. Okay, what's present at synovial, all joints is also synovial tissue. So people thought that maybe synovial tissue was responsible for synovial sarcomas. But it turns out that's not the case. So, but keep in mind that these cancers always arise near joints. That's a hallmark of those particular cancers. And they're complicated in the final, the pathology of the cancer is shown at the very last slide. And what we call histological, that is simply taking a blade right through a cancer and looking at what the cells look like. It has a complex histology, and I'll come back to that. It's called monophasic, biphasic, and poorly differentiated. So there are multiple histologies associated with that same tumor. Now, in this case, the gene that was newly created involved a gene called SYT, and that's on chromosome 18. And the other one is SSX. And there are 6 SSX genes in our genome, 1, 2, 3, 4, 5, 6, and they're all on the X chromosome. So the chromosome that if you have 2 copies, you're a female. If you have only 1 copy, you're a male with a Y chromosome. Okay, so you break, so this is the SSX gene, and this is the SYT gene. You break those 2 and form a new gene, shown here, and it contains almost the entire SYT gene and half of the SSX gene. And these genes are important for what we call chromatin remodeling. They're important for making the conformation of DNA in such a way that it will function either by turning genes on or turning them off. Okay, and what we call them are chromatin remodelers. And the new gene, and it turns out SYT is involved in complexes that are required to turn on genes, activation complexes. SSX is involved in different complexes that turn genes off, repressive complexes. The new molecule is a schizophrenic molecule. It has an activation domain and a repressive domain, both on the same molecule. And so you can imagine that this is going to cause trouble. So normally these genes, this one complex would be involved in turning genes off, the one off, the other one on. Now you make a new molecule that can do both. Okay, and that's going to raise a lot of havoc. So that's this gene. Yes?
Excuse me, how does the breakage you are talking about occur?
Occur. Good question. So the question is, how does the break actually occur? It turns out that in our cells, every day, every one of our cells has many, many breaks, 10,000 breaks. And they occur, for example, if you go out in the sunlight and sunlight hits on your DNA, it breaks it. That's one way. The other way is that you have factories in your cells, which are called mitochondria, that make a lot of energy. They have waste products. Those waste products cause DNA to break. So 10,000 times a day, you have these breakage and rejoining. Very, it's going on all the time. So it's no wonder that you could actually generate chromosomal breaks and rejoining with the wrong partners frequently. And the frequency is 1 in 1,000. Now, in our lifetime, we have about 10 to the 17th cells. Okay, that means 10 to the 14th translocations. Some of those are going to be bad, and some of them, like this, will give rise to cancers. So that's a good question. So they're just occurring all the time as not because we want them, but simply as a consequence of being alive. Okay, so this is going to be a little complicated. I told you we're not modeling it like breaking and joining. Okay? So we have to model it in some other way. And so what we do is take advantage of a system called LOXP. LOXP are 34, a sequence of DNA, ACTGs, that are 34 base pairs long and a very specific sequence. Okay, and if we take two of those sequences and we can do that, we can place them anywhere we want in the genome, and we flank a piece of DNA with those two LOXP sites. Then an enzyme, CRE, comes along and will mediate an exchange between those two sequences, and in the consequence, loop out everything in between. Okay, so by controlling where CRE is expressed, we can loop out a piece of DNA anytime we want. Okay? So that's our CRE-LOX system. And here's now what we made. We engineered in a cell to be able to then make that fusion gene that I just talked about under our control. Okay? And particularly under the control of CRE. So here you see SYT-SSX2. In this particular case, I should always be saying, I always say we. It's always the royal we. In this case, it's a very talented graduate student in my lab called Malay Halder. So he isolated this fusion gene from a human cancer, and it happened to have SYT-SSX2. So he has that piece of DNA in his test tube. And then he places it in a locus that's always expressed in that gene all throughout your life. When you're born, the first cell division, that gene is on and it stays on in every one of your cells forever. Okay? So any gene we put into that locus now will be turned on, except we have, and that's called the promoter, PRO, and in front of that we put a stop sign, and we flank that with LOXP sites. Okay, so now by controlling where LOXP site, or where the stop sign is, putting CRE, we pull out the stop sign, and we turn on this gene and makes the fusion gene. Okay, is that clear? So that's our model. So then by controlling where CRE is made, we can turn on that fusion gene in any cell we want. Okay, and then ask what happens. Does it make a cancer or not? And that way we can then control where CRE is and test different cells and says, does this make the cancer? And if it does, then that's a cell that is capable of making this cancer. We turn it on another cell and can't, then we know that cell can't make that cancer in the presence of that fusion gene. So that's the way we make in controlling and making that cancer. Okay? And then the other thing we do is put on a gene from a jellyfish. Jellyfishes have all sorts of beautiful fluorescent colors, and so we can isolate that gene that makes it green or red or yellow or purple, put it into our construct, and wherever now Cree is, those cells will turn green or purple or red or whatever we want. Okay, so we can then see the cancer because it's glowing